Okay, so what we need to talk about next is methods of increasing breeding success. This is important because breeding success is often lower in captivity. So we need to have a range of techniques to increase success rates. And one of them is cryopreservation. So this is preservation in cold storage, basically cold storage. And this is where we use liquid nitrogen typically to store things like eggs, sperm, or even embryos for a very long period of time. It's easier to transport. And it means that you can keep samples of genetic or reproductive material for a long period of time, even after the original donor is dead. So you can use like an egg or a sperm to fertilize something in the future to ensure that you have as wide genetic diversity as possible. Sort of related to this is artificial insemination, which is often abbreviated to AI. This is a technique that's widely used in agriculture, and it's where you take donor sperm and add it to an egg. Usually inside a female. So it's not in vitro, it's in the actual animal itself. And this means that you can avoid a number of problems that might otherwise occur. So you can avoid transport of large or like animals, or transport is inherently dangerous for animals as well, so it avoids harm. Mating can be dangerous. So it avoids things like injury or death, and this is especially important in large animals, things like elephants or rhinos. They don't get on very well together, so you're sort of risking the potential mother or father, basically, if you put them together. Putting potential, it avoids risks when trying to partner. So there's a case where ocelots, which is a type of cat, have killed potential partners. So really trying very hard to get these things to breed, keep introducing a new male into a female enclosure, and the female keeps killing the males. So I think there was 3 that were introduced to this ocelot, and they all got the chop. Next one then, again sort of related to this, is embryo transfer. Which is where we can take an embryo Well, let's start. So we start with we have few females left, or they have slow reproductive rates. We can treat with hormones to get lots of eggs at once, which are then collected. So they're like washed out of the uterus in a sort of operation. They are then fertilized and implanted into a surrogate mother. And typically the surrogate mother is a more common species. So there's potentially lots of them that we can use for this. And so you can, as a benefit of this, increase the number of offspring produced by one mother, because you're basically using her as an egg donor. You're getting the eggs fertilized, implanting them into surrogates. So you can, one mother might be able to basically produce dozens of offspring in a year as opposed to just one. The example for this one that you need to know is the, let's make sure I say this, bongo antelope. And they use the common eland as the surrogate. Which is a different type of relatively closely related antelope. Another one we can do is micropropagation. This doesn't work for animals, I'm afraid. We have to do this for plants. And this is where we take a tissue culture, in effect, a type of tissue culture. So that's like a bunch of cells, typically from the meristem of a plant. You put them into a culture, or like a gel, basically, in a petri dish, and encourage them to develop into individual plants. So you have many genetically identical plants from one parent. So we can rapidly increase a population. There are issues with this because they're all genetically identical, so that's not so great. And then the last one is cloning. And this is an interesting one because you take an embryo and replace its DNA with DNA from adult stem cell. And then it's implanted into a surrogate mother. This has not worked very much. That's due to a number of reasons. It could be the damage from deep freeze. Might have caused the DNA of the stem cells to be less viable, but it's potential that it could be used in the future to either recover functionally extinct organisms, so there's only one or two individuals and they're the wrong gender or something like that. So something like the northern white rhino. Where this is possible. But it is possible to use for plants. So we can use it for things like the Atlantic black poplar, which is a UK species of tree, which unfortunately very easily hybridizes with introduced subspecies. So if we want to keep a pure gene pool of the population, we use cloning to produce identical young plants from non-hybrid parent plants. So it does work for plants, but not for animals yet. Okay, so there might be woolly rhinos or woolly mammoths on the horizon eventually, I guess.